Okay, we're doing lesson 50 today, but I'm looking right now at lesson 49. And we talked about codominance and X-linked traits. And the example I provided was fur color in cats. With some breeds of cats, fur color is an X-linked trait. You see that a female can be homozygous black, homozygous orange, or she can be heterozygous and be black and orange. We call that color pattern calico. It's also been stated that every mammal, every organism, has all of their chromosomes in their body cells. So, John, if I took a cell from the tip of your nose, how many chromosomes would I find in that cell? And you're a male, so what sex chromosomes would there be? So you have sex chromosomes in your nose. Yes, you do. Rachel, if I took a cell from the tip of your nose, how many chromosomes would I find? And what would your sex chromosomes be? So you have two X's in every cell. That is correct. And so when it comes to these calico cats, they have an X chromosome with black fur and an X chromosome with orange fur. And I have a picture of a calico. Let me bring that up. There she is. If you guys take a look at this cat, I want you to look at the cat's shoulder. What color does this cat's shoulder appear to be? Orange. Well, yes or no? Does the cat have both X chromosomes in the cells in that shoulder? Yes. Then why is it just orange? Why isn't it orange and black? And conversely, look at the midsection of that cat. It's black. Does that cat have cells with both X chromosomes in that part of his body? Yeah. So why in the world is it just black and not orange and black? Well, to answer that question, it gets me to the third topic that we discussed, which leads me to today, and that's called X inactivation. So with you two, Rachel and John, I want to ask you some questions again, okay? Rachel, how many X chromosomes do you have in every cell? You have 2. John, how many do you have? That's not fair. Rachel has 2, John has 1. So feasibly, Rachel is getting twice the X-linked trait expression that John is. Males and females have to be compatible. And so in every mammal, I should say, one of your 2 X chromosomes is shut down. The proper term is methylated. Let me be clear that those chromosomes are there. They're present, just like a lot of your cars are present in the parking lot, but they're off. That X chromosome that is turned off is called a bar body, and it is methylated. So the example I have with this ugly-looking cat here, I want you to look at the orange part of this cat's face. Listen to my questions carefully. Does that cat have 2 X chromosomes in every cell on the part of its face that is orange? Yes. Which X chromosome is the bar body on the orange part of its face? Is it the X chromosome carrying the orange gene or the X chromosome carrying the black gene? Black. So what's the only trait that's left to express? Orange. And conversely, does that cat have 2 X chromosomes in every cell that's on the black part of its face? Yes. Which X chromosome is the bar body, ergo shut down, methylated? The orange one. So what is the only trait left? Black. This is known as methylation. And it can be undone, which is what we're going to get to today, but it prevents the overexpression of X-linked traits so males and females can have a level playing field, so to speak. So I'll give you guys a moment to jot down the title of your lesson for today, and only today. This is a good Monday lesson. We get out early on Mondays. Epigenetics and epistasis. I'll give you a moment to jot that down. If you're at home, just hang on a second. Some of you told me that earlier in your life you were blonde, and now you're brunette. That is epigenetics. Highly likely that is epigenetics. I'm not going to say 100%, but it's highly likely it's epigenetics. So in epigenetics, these are factors that determine your phenotype beyond your genotype. Typically, when you see a genotype, you've been able to tell me the phenotype. But now I'm going to say these are going to be influences beyond your genotype. So it is inheritance, influenced by factors beyond the nucleotide sequence. And you're going to hear all about that in Unit 6. Your nucleotide sequence is your literal genetic code. Adenine, guanine, thymine, cytosine, A, T, C, G, G, T, C, A, T, G, C, T, all that stuff. That's your genetic code. This is essentially your genotype. So I'm going to pose a question, you guys, I asked a lot a few months ago. When you were a baby in a diaper, pooping your pants, completely dependent on your parents, did you guys have the genetics to grow armpit hair? Yeah. The genes are there. Were they turned on? No. So we would say that they were methylated. But now you all are teenagers. You've been through puberty, most of it. It's been demethylated. The genes are now expressed. So I want to remind you that just because you don't show a trait doesn't mean it isn't there. It just might be covered up. It might be masked. So the example I want to give here today is going to be a dominant allele is going to equal brown hair. And a recessive allele is going to equal blonde hair. Okay? If you were to perform a genetics analysis of somebody's DNA and you saw that genotype, homozygous dominant, what would you conclude their phenotype is? Brown. Sure. You would have no reason not to. What if you saw this genotype, homozygous recessive? What phenotype would you conclude they have? This is not a trick question. Blonde hair. And you would have no reason not to. What if they were heterozygous? And this is, by the way, complete dominance. That's heterozygous. What color hair would you expect them to have? Brown. And you would have no reason not to. But what if I had this person and I look at their DNA and it says heterozygous, but I look at them, my eyes work just fine, and they're blonde? What gives? Here's what's happening. One of their alleles might have been methylated. And I'm going to come back to that little smiley face person in a moment, but I want to start back with the homozygous dominant individual, okay? And I want you guys to see how this works. When a gene is methylated, it is present. It is just shut off. It's covered up. You can't access it. What if that allele was methylated in that genotype? If you missed what I just did, watch again. Oops. Watch this. That first dominant allele is methylated. What is the only other allele that they have to lean on? A dominant allele. So what hair color would they have? It would still be brown, right? All right. Round two. What if this allele was methylated? Same thing, okay? Let's look over here at this blonde person. What if one of their recessive alleles was methylated? What would their hair color be? Okay. What if this one was methylated? What would their hair color be? That's all they've got. Now let's go to the heterozygote. What if their dominant allele was methylated? What color would they have? Blonde. And let's say later in their life, 5 years old, 7 years old, maybe 12 years old, that methylation goes away and the gene can now be expressed. What hair color would they have? It would change to brown. That's right. Okay. Now, how about this? What if this allele gets covered up? What hair color will they have? Brown. And what if the methylation goes away? What hair color will they have? still brown. So you have to really pay attention to the details on this and which alleles are being covered up. So for those of you that used to be blonde, this was likely you. And you were a blonde kid. And now that went away. And you're no longer blonde. You are brunette. Yeah. Not necessarily, you know, let's say that I know for a fact that I am heterozygous. My mom was blonde. And let's say when I was a kid, my blonde allele was methylated. What hair color would I have? I would have brown hair. And I've always had brown hair my whole life. And let's just say that later in my life, the methylation went away. Even though the methylation went away, what hair color will I still have? Brown. So there's really no way to tell, Cyrus. The only thing that you would know is if I were to test your genetics, and if I saw this genotype and it says heterozygous, that means brown, but I look at you and you're blonde, that might explain something about you do in fact have a methylated gene, and it hasn't been demethylated yet.
Which one, like, is there some factor that determines which one will be methylated?
Random. Yep. Sometimes it can be environmental, other times it can be inherited. Okay. And now, speaking of that term methylation, I want to talk about that. Why is it doing this? I don't know why it's writing all crooked for me, guys. Hold on 1 second. All right, I'm just going to try to spell this out. I don't know why the letters are shifting on me like that. Here we go. When a segment of a gene is methylated, you cannot imagine how confusing this is to write. It cannot be accessed by transcription factors. And therefore, now we want to write straight, cannot be expressed. So your gene for armpit hair was there the whole time, but your ability to express it was inhibited. Couldn't get to it. It's like trying to find that quarter that fell between your laundry machines. It's there, you can't reach it, so you can't have it. I don't know if this ever happened to you. You got a little quarter, it falls right between your washer and dryer. It's there, you just can't get to it. That's what happens with these genes that are methylated. It's there, you just can't get to it. But sometimes the methylation goes away, and we call that acetylation. So this can be undone. When something is acetylated, you can say it's been demethylated. All right. That's it for epigenetics. Let's get to the other topic. That's epistasis. Have y'all ever known someone that's had brown hair on their head, brown eyebrows, but they have an orange beard, like this guy? You ever seen someone like that? I'm going to tell you why that happens. So, epistasis. Is a circumstance where the expression of one gene is modified, and in that term, it typically means masked, inhibited, suppressed by the expression of one or more genes. That's kind of a complicated definition, so I'm going to shore that up a little bit. You guys have traits, and they influence your phenotype. But sometimes those traits are influenced by a higher power, so to speak, like a puppeteer. And that will influence whether your genotypes will be expressed normally or not. So to try to explain this, I know this one was always really tricky to try to teach. I mean, I get it. I know it, but I need to make sure you guys know it. So the example I want to get is llama fur color, okay? Y'all know what a llama is? It's kind of like a camel. They live in South America. They spit. They're a mammal. They're very furry. Have a lot of wool. In llamas, a dominant allele will be black and a recessive allele will be brown. So let's start off easy. If you have a llama that's homozygous dominant, what color will it be? If you have a llama that's heterozygous, what color will it be? And it is complete dominant. Black. And if you have a llama that's homozygous recessive, what color will it be? Easy enough, right? Easy enough. Well, when it comes to fur color, the bees, whether they be dominant or recessive, are controlled by a more influential gene. And we're going to call these the epistatic genes. This is going to be normal. And this is going to be mutated, which will result in no pigment, which is generally albino. So, here's the catch. As long as one dominant D exists, it's status quo. That means that everything is going to be as it should. Homozygous dominant, black, heterozygous black, homozygous recessive, brown. So I'm going to go through all the genotypes I can think of, and I just want you guys to tell me what color the llama would be, okay? And I'm going to help you out a little bit. Here we go. First question, what is the genotype of the bees? Homozygous dominant. We got 5 minutes, so I need you guys to pick it up. Do you see any dominant Ds in that genotype? So it's status quo. So what will the color be? Black. Great, and we're off. All right. What is the genotype for fur color? Heterozygous. What do you see any dominant Ds? So status quo. What's the fur color? Great. What is the genotype for fur color? So are there any dominant Ds? So status quo. What's the fur color? Great. What's the genotype for the fur color? Homozygous dominant. Do you see any dominant Ds? All right, status quo. Black. All right, now let's get to this. What is the genotype for the fur color? Homozygous recessive. Do you see any dominant Ds? So what is the fur color? Brown. What is the genotype of the fur color? Homozygous recessive. What is the or do you see any dominant Ds? So what's the fur color? Great. Now let's check this out. What if you have this and you have that? What is the genotype of the fur color? Homozygous dominant. But do you see any dominant Ds? The Ds control the Bs. That's epistasis. And whenever you do not have any dominant Ds, you're going to have the mutated form, which is basically albinism, no pigment. So despite the fact that you have homozygous dominant black, that is being controlled by another trait. What will the color of the llama be? White. Because there's no dominant Ds there. What is the genotype for fur color here? What color would you think that would be? That would be black, but because there's no dominant Ds, what color will it be? It'll be white. And then finally, what fur color will that llama have? White. So this is all about the D genes are influencing the B genes. When you have a person like this, they have a normal MC, is it MC1R or MCR1? MC1R. They have a normal MC1R gene, but here they have a mutated MC1R gene. If you have one copy of a mutated MC1R, your brown hair on your beard will go orange. If you have two copies of a mutated gene, you're going to be redhead top and bottom. All right, here's what I'd like you guys to do tonight. You don't really have any take-home homework. You set work on your review and your mastering. I want you to cross a heterozygote with a heterozygote. I want to know all genotypes and phenotypes and phenotype ratios. And I'm going to tell you now, it is not going to be 9-3 to 3-1. It's not going to be that easy. If it was that easy, I wouldn't give it to you. So do a dihybrid cross and bring those tomorrow.